Hello, and welcome back to Lecture 5 in the Foundations for Kinematic Analysis lecture series. In the previous lecture, you learned how to extract motion tracking data from videos. We consider this the raw motion tracking data. Today, we will show you how to clean and process the data to get it ready for kinematic analysis. Data preprocessing is the process of transforming raw, messy data into a format that is suitable for analysis. Our goal is to reduce noise from tracking inaccuracies and deal with the variability in our videos that makes it hard to compare across participants. We do this in two steps. The first step is smoothing, which gets rid of jitter, noise, and other artifacts. The second step is normalization, where we bring all the participants to a common scale in a standardized coordinate space. This is an example of one of the videos we extracted in the previous lecture. The motion tracking data displayed with the skeleton overlay looks pretty good already, but if you focus on any landmark, you'll notice that some points jump around a bit. This is especially apparent in the hips and fingers. We call these frame-to-frame fluctuations jitter. We want to get rid of this noise so that the movement is smoother, matching more accurately to how our participant is actually moving. There are a lot of potential sources of jitter or noise in our data. The MediaPipe body and hand models we used are not perfect. They are an estimation. So there is some frame-to-frame prediction and accuracy and inconsistency. Another source of jitter comes from occlusions. This means that the camera can't fully see a body landmark. In this example, the speaker's hand passes behind a microphone, but this can also happen when one hand passes behind the other. Another source of noise is motion blur from fast hand movements. If the model can't see the hand clearly, it's hard to estimate the landmark positions. The next example is bad lighting. This gentleman on the bottom is covered by a lot of shadows, which makes it hard for computer vision models to identify body parts. Similarly, having a lot of background clutter can make it difficult to distinguish the speaker from the background. These are all things we discussed in Lecture 3 when thinking about how to optimize your data collection for video-based motion tracking. It's great if you can avoid as many of these issues as possible when collecting your data, but it's impossible to eliminate all sources of noise in the data. Smoothing will help us reduce the impact of that noise. So why do we need to smooth our data? It's not about making our data look nicer. It's about recovering the underlying continuous movement signal from a noisy measurement process. Smoothing improves the biological plausibility of our motion data. Extremely rapid fluctuations in wrist and finger positions are unrealistic and do not reflect actual human movement. Although the data may not look too noisy now, this noise actually gets amplified later when we derive kinematic measures like velocity and acceleration. The conditions of the videos also differ across participants. Participants may have different lighting, clothing, and backgrounds. Smoothing reduces these external sources of variation. Finally, a smooth movement signal ensures that statistical differences reflect true movement differences rather than measurement error, and makes movement patterns easier to interpret visually. Before we start smoothing our data, we first have to handle any missing data. If you look at this video clip, you can see that the speaker's finger tracking from the MediaPipe model come in and out of detection. You'll also notice that the wrist from the MediaPipe body model has a lot of inaccuracies and seems to confuse the speaker's hand with the pattern on her shirt. If the sources of noise are significant enough and visibility is low, the model will fail to detect the landmark at all, and this results in missing data points. In our case, the missing data is mostly in the fingers. So first, we need to get a sense of how much missing data we have for the landmarks we are interested in. If your video data are decent quality, you will likely only have short gaps of missing data. If that's the case, we can use linear interpolation to fill in the gaps. Linear interpolation takes the value before the gap and the value after the gap and draws a straight line between them. So in this plot, you can see the motion tracking data for the right index finger before linear interpolation. The horizontal position of the finger is plotted on the y-axis, and time is plotted on the x-axis. And you'll notice that there are a few small gaps in the motion signal. This is what the plot looks like after interpolation. You can see the orange lines have now filled in the gaps. These were all relatively short gaps in time, maybe up to 5 frames at our 30 Hz sampling rate. However, if you have long gaps of missing data, it's better to exclude those segments from data analysis. So the result might look something like this. The video on the left is the same example you saw previously with missing finger data. The video on the right shows the result after using linear interpolation to fill in missing points. The result isn't perfect, but it's closer to reality than before. After linear interpolation, we're ready to smooth the data. There is not one standardized way to smooth your data, but in general, we want to apply some kind of filter. Let's think about our motion tracking data in terms of frequency. Frequency tells us how fast something oscillates or changes over time. In our case, this refers to how quickly a body part's position changes from moment to moment. An example of a low frequency movement would be a slow, smooth movement like raising your hand. An example of a high frequency movement would be rapid, small fluctuations. For example, if your hand is trembling. Most intentional human movements like walking, reaching, and gesturing happen at low frequencies, producing smooth and continuous movements. This plot shows a zoomed-in frequency spectrum of the left index finger of one of our participants, with frequency on the x-axis. Here you can see that most of the energy concentration is at low frequencies, less than 2 Hz. In contrast, the noise we get from tracking inconsistencies are rapid small fluctuations and random movements that are much higher in frequency. Our goal is to separate the signal from the noise. To do this, we will use a type of low-pass filter called a Butterworth filter. Low-pass filters allow low-frequency signals to pass while attenuating high-frequency signals. One of the most important parameters is the cutoff frequency, which is indicated here by the dashed vertical line and abbreviated FC. This defines the boundary of our pass band and our stop band. This is the frequency where the filter starts to noticeably reduce the signal at about 70% of its original magnitude. This number is not a hard cutoff. From here, we have a smooth, gradual attenuating function. The steepness of this function is related to filter order. We'll be using a filter order of 2, which is more gradual. In contrast, a higher order would more steeply attenuate frequencies in the stop band. So the next question you might be asking is, how do we determine the right cutoff frequency? In general, you may want to consider a value less than 10 Hz, since we know that biological motion tends to be lower in frequency. We will use a data-driven approach to further narrow it down. To do this, we'll try out many candidate cutoff frequencies and plot the results. In this plot, you see a continuous range of cutoff frequencies between 0 and 10 Hz on the x-axis. And on the y-axis, we're plotting the residual. Residual means the difference between our original signal and our filtered signal. So at low cutoff frequencies, the filter is aggressive, smoothing out almost everything. And this means that the residuals are high because there is a big difference between the smoothed and the raw data. As we increase the cutoff frequency, we allow more signal through, so the residuals decrease. We want to pick a cutoff frequency where the residuals begin to plateau, and we call this the noise floor, which is defined by the intercept. This gives us a recommended cutoff frequency of 2.88 Hz in this example. So in this case, we would apply a second-order Butterworth filter with a cutoff frequency of 2.88 Hz. After applying the filter to our data, we can plot the raw motion tracking data and the smooth motion tracking data. So in this case, the raw data is in orange and the smooth data is in green. But this is hard to see, so let's zoom in. Now we can see that our smoothed curve follows the general pattern of the data, but we've gotten rid of some of the sharp, kinky edges. And this is what we want to see. We're maintaining the overall shape of the signal, but we're getting rid of the noise. We can further inspect the results by viewing the skeleton overlay videos before and after smoothing. So here we see quite a lot of jitter in the hands before smoothing, but the landmarks are much more stable and smooth after we've applied our Butterworth filter. It's important to avoid the danger of over-smoothing. For example, the black line in this plot shows what the smooth data would look like if we used a very low cutoff frequency of 0.5 Hz. You can see that this black curve completely wipes out some of the nooks and crannies of our motion signal, but these bumps are not meaningless. They are the motion signature of our gestures, showing the pattern of how the index finger moved left and right during key gesture events. When we over-smooth, we risk washing out the very signal that we're trying to capture and understand. And this is why it's important to use principled cutoff values and check the effects of smoothing on your raw data. Indeed, if we compare the skeleton overlay videos for the optimal cutoff frequency on the left with the over-smoothed data on the right, we can see that over-smoothing has completely changed and reduced the signal. So whereas optimal smoothing improves the biological plausibility of the data, over-smoothing can wipe out the signal. The Butterworth filter that we used in this lecture series is commonly used in biomechanics, but it's not the only type of filter you can use to smooth your data. These are just a few other options you can consider. All of these options use a moving window approach. The moving average filter replaces data points by averaging across short time windows. The Savitsky-Golay filter, which is pictured here, uses a low-order polynomial with a sliding window of data. And the Gaussian filter uses a bell-shaped weighted moving average, where nearby points are weighted more than distant ones. So it's up to you to make your own decisions around smoothing your raw motion tracking data, but we hope that this information will help. Smoothing was step 1 of pre-processing our data. Now it's time to move on to normalization. Let's start by taking another look at our 8 participants. We have a lovely group of participants, and there's quite a lot of variability between speakers. Some of this variability is due to the speakers themselves. People come in lots of different shapes and sizes. We all have different arm lengths, hand sizes, and shoulder widths. But there are also a lot of differences in the recording setups. Some speakers are very close to the camera, while others are farther away. Some speakers are captured at a direct, straight-on angle, while others are angled slightly to one side or the other. These differences can make it difficult to compare their gestures. The goal of normalization is to get everyone on the same scale. In research, we often want to compare behaviors across participants. This is especially critical for analyzing the spatial properties of gesture. For example, how large a gesture was or where it was produced in space. It may also be important for training machine learning models so that these models focus on learning the properties of gestures themselves rather than the differences in recording variation. You might wonder whether normalization is always necessary. This really depends on your research question. In general, it may be less critical if you're looking at within participant changes only, if you're focusing on temporal properties like timing or frequency, or if you're analyzing relative motion, for example, wrist position or velocity relative to the shoulder. But you may still find normalization useful even in these cases. There are a few different changes or methods of normalization you might consider applying to your motion tracking data. We're going to go through each one in more detail. The first change we'll make is adjusting our motion tracking data to match the aspect ratio of our video and to invert the y-axis values so that larger values of y correspond to positions that are higher in space. We will also normalize the size across all of our participants. Of course, people come in all different sizes, but size differences can also be due to differences in camera distance. There are also differences in how participants might be positioned in the camera frame, so we want to bring everyone to the center. And there might be variability in how participants are oriented due to differences in camera angle. This one is a little bit more difficult to correct for, but we'll give you some tips. As you can see, if we take any two participants and plot their raw, unnormalized skeletons in coordinate space, they don't overlap very closely. The goal of normalization is to make these skeletons as similar as possible across participants. For the first step, we're going to correct the video's aspect ratio. Remember that MediaPipe produces values between 0 and 1 for both the x and the y axis. However, our videos are not perfectly square. Because of that, our x-axis gets compressed relative to our y-axis. This means that using MediaPipe's default scaling, horizontal movement is weighted less than vertical movement. Another thing that is not intuitive from MediaPipe's default output is that higher values on the y-axis correspond to positions that are lower in space. You can see here that the top of the frame corresponds to 0 and the bottom corresponds to 1. This means that even though the bottom participant's wrist is higher than her elbow, the y value of the elbow is larger. To address these issues, we first inverted the y-axis. We multiplied y values by -1, so now instead of ranging from 0 to 1, it ranges from 0 to -1. Next, we multiplied the x values by the aspect ratio. For example, our videos have a resolution of 1920 pixels wide by 1080 pixels high, creating an aspect ratio of about 1.78. When we multiply the x values by this number, we see that our x-axis now ranges from 0 to 1.78, accurately reflecting the original aspect ratio of the video. The next issue we'll address is the size of the participants. Because one participant is closer to the camera than the other, their bodies appear to be different sizes. We want to make everyone's torso the same length. To normalize size, we're going to use 4 reference landmarks on the body: the left and right shoulders and the left and right hips. For each video frame, we find the position of the participant's neck by averaging the x and y participants of their left and right shoulder. Similarly, we find the position of their mid-hip by averaging the x and y positions of their left and right hip. Then, to get the torso length of each participant, we calculate the Euclidean distance between the neck and the mid-hip using this formula. Euclidean distance is the straight line distance between any 2 points. So here, we calculate it as the square root of the sum of the squared differences between the x and y coordinates of the neck and the mid-hip. Although we are normalizing participants' size using the torso length, You could also normalize based on other joint measurements, for example, the distance between the two shoulders. So we want to make the torso lengths of all participants equal to 1. For each individual participant, we take 1 divided by their torso length to calculate that participant's scaling factor. Then we multiply the x and y values by that scaling factor to get everyone on the same scale. After size normalization, the skeletons look something like this. Now we can see that the torsos of our participants are the same size, both with a torso length of exactly 1. This allows us to easily compare the size of the two participants' gestures. However, if we want to compare the height or location of their gestures in coordinate space, we also need to align them to the same position. To normalize position, we are going to translate the skeletons and pin the neck of each participant to the origin. This means that the coordinate values for the neck of all participants should be x equals 0 and y equals 0, and here it's indicated with the star. Because the torso length of all participants is 1, we see that the mid-hip value is now at y equals -1. By normalizing size and position, we reduced a lot of variability in our data to make it easier to compare participants. For example, we can now easily and confidently say that the right wrist of the orange participant is higher than the right wrist of the green participant, and we can quantify by how much. This result looks quite nice, however, there are a couple more things to consider. The example I just showed you applied size and position normalization to a single video frame for 2 participants. One option is to apply this normalization to every single frame in the video. This ensures that in every video frame, the participant's torso length is 1 and their neck is at the origin. This is the approach you see on the plot on the left. This plot shows frame-by-frame normalization for all 8 participants in the dataset. I chose a random frame about 28 seconds into each video, and you can see that the normalized skeletons are still beautifully aligned. In contrast, in this plot on the right, we calculated the average torso length of participants across all video frames and the average position of their neck, and we normalized based on these values. This means that, on average, the participants' torso lengths are 1 and their neck is at the origin, but there is some variability from frame to frame. If we look at the same time point in this plot, we see that the skeletons are a little bit messier. These two methods have different advantages. Even though the left plot is cleaner, there are good reasons why a participant's torso length might vary from frame to frame. For example, even if I'm sitting and in a relatively fixed position, I can still lean forward with my body and that will shorten my torso. Or I might reach with a gesture and shift my torso to the side. If you restrict participants to the exact same size and position in every single frame, you lose some of this natural variability and posture shifts. For this reason, in this lecture series, we chose the scenario on the right, averaging across frames to allow for some of this natural variation. That said, if your participants move or drift too much, or you need a really consistent body reference point for your analysis, you might prefer the scenario on the left. This lecture is not meant to be prescriptive to tell you exactly how to normalize your data. That will depend on how much variability there is in your recording setup across participants and what you're trying to measure. But regardless of the methods you use to normalize your data, we recommend visualizing the results and checking that the normalization did not distort the movement characteristics you're interested in. Finally, we normalized the size and position of participants, but we did not account for differences in body orientation. In our dataset, we have a couple of participants who were not captured from a straight-on angle, so their bodies are slightly rotated. This is not ideal because it means one arm is closer to the camera than the other, and their movements are not symmetrical. In order to correct for rotational differences and body orientation, you need reliable 3D motion tracking data. You can do this with markered 3D motion capture systems like Vicon, or you can do it with video-based motion tracking if you triangulate 3D motion data from 2 or more synchronized cameras. For this lecture series, we were not able to correct for these orientation differences, so this is a limitation of our dataset. Now that you've learned the ideas behind smoothing and normalization and seen some examples, it's time to try it for yourself. Let's move on to our Jupyter Notebook tutorial. Okay, let's navigate to the Lecture 5 script folder. Open up the Jupyter Notebook file. Like before, once you get that open, you're going to click Select Kernel in the top right corner and choose your Python environment for the course, KLS environment. In the previous lecture, we extracted motion tracking data from videos using MediaPipe. After our last notebook tutorial, we ended up with CSV files containing the X, Y, and Z positions of body landmarks over time. By the end of this notebook, we will have filled in small gaps of missing data, smoothed out high-frequency noise in our motion signal, and normalized participant size and position so that we can compare data across participants more easily. Just like before, I won't be explaining every line of code. Instead, I'll give you a general overview of what each section is doing. This first section is about handling missing data. First, we're going to run the cell to import the packages and tools that we need for this notebook. Next, we'll read in our motion tracking data. In the previous lecture, we saved 8 CSV files in the Output Time Series folder of Lecture 4. We are defining the path to that folder. Then in the loop below, it looks for and reads in all CSV files that are contained within that folder. We are loading these CSV files into a data dictionary, where each file is stored with the key name that we can refer to. You can run this now, and then you'll see a list of the motion tracking files that were successfully loaded in the output. Before cleaning the data, it's important to get a sense of how much missing data there is and what the missing data actually look like. In the next plot, we visualize the X and Y positions of the left index finger over time for one participant. Go ahead and run this cell. In the plot below, you'll notice that there are occasional gaps in the data. These missing values can occur when MediaPipe temporarily loses track of a body part. The videos in our toy dataset aren't super high quality recordings, so we do lose some data, especially in the hand tracking, because of motion blur or poor contrast. These gaps are usually quite small, which means that we can use linear interpolation to fill them in. In this next cell, we define our function called cleanData to perform linear interpolation. The basic idea is that we estimate what happened in the missing gap by drawing a straight line between the tracked points immediately before and after the gap. Go ahead and run this cell. It will loop through all the motion tracking files in our data dictionary and interpolate any missing data from our motion tracking data columns. Then we plot the result below, and you can see that all of the missing gaps have now been filled. In section 2, we will smooth the data. Our goal in this section is to get rid of high-frequency noise in our motion tracking data that results from small frame-to-frame fluctuations. We want to remove this tracking noise while preserving the overall motion pattern. In this next cell, we define a function for a Butterworth filter, which has parameters for the data we are filtering, the sample rate of the video, the filter order, and cutoff frequency. You can run this now. One challenge is deciding what the cutoff frequency should be, or in other words, how much smoothing to apply. Too little smoothing leaves noise in the data, and too much smoothing can remove real meaningful movement. In this cell, we are manually exploring the effect of different cutoff values. Here we've defined the order as 2, which is a gradual smoothing function. And we've defined two different cutoff values, an 8 Hz cutoff frequency and a 0.5 Hz cutoff frequency. We'll apply our Butterworth filter using these two different values and then plot the results. The unfiltered original data is in black. You can see that the cutoff frequency of 8 Hz did not affect the data very much, whereas the 0.5 Hz cutoff really changed the shape of the signal. The nice thing about this plot is it is interactive, so you can click on the different traces in the legend to remove them from the view, or zoom into a specific section to inspect it more closely. Rather than guessing and trying out different cutoff frequencies manually, we're going to use a data-driven approach to determine how much smoothing should be applied. In the next cell, we define a function called compute residuals versus cutoff. Basically, we give this function a bunch of different possible cutoff frequency values, and it applies the smoothing filter at each frequency. Then it measures how much the smooth signal differs from the raw signal, and this difference is called the residual. For each cutoff frequency, it stores that residual value. Then it looks at the last half of the residuals and fits a straight line through them to represent the noise floor. The optimal cutoff frequency is determined by the point where the residual curve meets the noise floor. We're going to decide what landmark we want to use to estimate the optimal cutoff frequency. I'm using the right wrist because the wrist is the primary landmark we'll use in our analysis. And then we specify the range of cutoff frequencies we want to try. We are going to estimate 50 cutoff frequency values that fall between the range of 0.5 and 10 Hz. Then we will loop through all 8 of our saved motion tracking data frames and run each one through the compute residuals versus cutoff function. We want to visualize the frequency response curve for each video, as well as the average values across videos. So we'll plot both results. Let's run this cell now. The gray lines represent the curve for the individual videos, and the thick green line is the average curve. The dotted vertical line represents the recommended cutoff frequency where the residual curve meets the noise floor. In my case, the recommended cutoff frequency is 3.34 Hz, but you might have a slightly different number because each time you do the MediaPipe landmark extraction, you'll get a slightly different estimation of the coordinate locations. In the next code cell, we will apply the Butterworth filter to all 8 of our motion tracking files using the mean recommended cutoff frequency that we calculated above. Go ahead and run this cell. The notebook will process each participant's data and create a new version where the motion tracking columns are smoothed. We'll save the new version to a subfolder called Smooth CSVs in our output folder. Next, we're going to visualize our smooth results. This plot is similar to the one above. We're going to choose one file and one motion tracking column, in this case, the X right wrist. And we're going to plot the original signal and the smooth signal. Let's run this one. And from this view, it's hard to see much difference, but let's select a portion to zoom in on. Now you can see that the smooth line in green follows the same overall movement pattern, but removes small fluctuations and jitter. Remember that at the end of Lecture 4, we created the skeleton overlay videos with the raw motion tracking data CSV files. If you want to see what your interpolated and smooth data look like on the original videos, you can run that code on the smooth CSV files to generate new skeleton videos. This is also a really helpful way to check that the smoothing improved the motion tracking accuracy. Okay, now it's time to talk about normalization. I'll run this first cell to read in an image from our screenshots folder. This image is just to remind you that people come in all different shapes and sizes, and that sometimes the camera recording setup isn't as consistent as we'd like it to be. In this case, our participants are also standing at different distances from the camera. This means that their motion tracking coordinates will vary a lot, and it's hard to compare them. Let's take a look at how the participants look in coordinate space before normalization. We're going to plot their skeletons, so I'm defining some joints and connections, or bones, that we're going to draw. We will loop through all of our smooth CSV files and visualize the participant torso skeletons from the first frame, which is indexed as 0. Let's run this now and look at the plot. You can see that the participants appear in different locations and at different scales. We want to normalize them by centering them all to the same position and scaling them to the same size. In the next cell, we define our normalize size position function. This function has a few parameters. Suffix refers to what the end of the column name says. So by default, the suffix is set to a blank empty string because my CSV files have both image-based and world body landmarks. And in this case, we're only going to apply normalization to the image-based landmarks, which do not have a suffix. By default, target height is set to 1. This means that after normalization, the torso length of all participants will be 1 unit. You can choose whether or not to invert the y-axis, but we recommend to do it to make it easier to interpret. So by default, it's also set to true. And then we will correct for the aspect ratio. The default is assuming an aspect ratio of 1, so a square video, but you can change it to your video aspect ratio. So in this function, first we identify the relevant motion tracking columns, then we invert the y-axis, then we correct for the aspect ratio by adjusting the x motion tracking columns only, then we calculate the torso height of each participant in all video frames by using the distance between the mid-shoulder and the mid-hips. We calculate the average neck position and the average torso length for each participant. Then for each participant, we calculate a unique scaling factor based on the target torso height, which is 1.0, divided by the mean torso length. Then we apply that scaling factor to all frames in our motion tracking data. As I mentioned in the lecture, this approach normalizes by averaging size and position across all video frames so that, on average, participants' necks are centered at the origin, and on average, their torso length is 1 unit. If you want to normalize size frame by frame, you would adjust this section so that you calculate a unique scale factor for each frame rather than for each participant. Okay, so that's just defining the function. Now we have to run the function. Our videos are not square, so we need to first define our aspect ratio. If you're unsure, you can look at the details of your video file. I'll pull up one of the videos. So we go to course materials and then input videos. On Mac, if I right-click on it and select Get Info, I can see under dimensions that my video is 1920 by 1080. So that's what I've put here. This corresponds to an aspect ratio of about 1.78, which is a standard 16 by 9 ratio. Now we will loop through all of our smooth data frames. We keep the settings we discussed, and aspect ratio is set to the aspect ratio we defined. Let's run this. Now it's nice to see the results after our normalization. Let's plot the participant skeletons again. We can pick any video frame. Right now I'm visualizing the 400th frame, which is about 13 seconds into each video. Let's run this. Now we can see that the skeletons are stacked much more nicely. But since normalizing takes the average size and position across all video frames, there is still some natural variation. Let's try picking a different frame. If I choose the 600th frame and run it again, you can see that the skeletons have shifted a little bit. Again, you need to decide for yourself, based on your research design and question, whether you want to allow this variability or if you prefer to normalize and constrain the size frame by frame. Next, we're going to save our normalized data frames as CSV files in a new output subfolder called Normalized CSVs. The motion tracking data in these CSV files has been interpolated, smoothed, and normalized, so they are fully pre-processed. But I do have a written reminder here. Remember that we only normalize the X and Y columns, so Z is unchanged. You can run this now. And then when I do data analysis, I prefer to have all of my participants' motion tracking data in a single file. So that's what we're doing in this last step. We're combining all participants' pre-processed data into a single master CSV file. And as I mentioned in the previous tutorial, we have a massive amount of motion tracking data, but we're only going to use a subset of these columns. So we're also going to filter out the unused columns to reduce the size of the data. We will keep only the motion tracking columns for the wrist, elbow, and index fingertip. So let's run this now. And then let's take a look at our output folder. The file we just created is called allnormalizeddata.csv. Let's open it. You can see that it already looks a lot nicer with fewer columns. And we also have a column called video, which is the name of the video we processed. In our case, this is also a unique name that identifies each participant. So it starts with the Pen Pals video, but if we scroll down, eventually we get to the Campfire Confessions and the rest of the videos. And that's it for this notebook tutorial. We did a lot of work to pre-process the motion tracking data, but now they are cleaned and ready for analysis. In this lecture, you learned how to pre-process your raw motion tracking data. We used linear interpolation to fill in missing values. We applied smoothing to reduce artifacts due to tracking inaccuracies. And we normalized across participants to correct for video aspect ratio and differences in body size and position. There were a lot of complex concepts in this lecture, so great job sticking with it. We are almost ready to begin with our kinematic analysis, but first we have one more step. In gesture research, we often work with annotations from ELAN that tell us when gestures occurred. In the next lecture, we'll show you how to merge multiple data streams together so everything is in one place. This will allow us to analyze the movement signatures of specific gesture events. So thank you for following, and we'll see you next time.